Thank you very much for the opportunity to talk to you today about an exciting project that we are working on here at the Jonsson Comprehensive Cancer Center at UCLA. My name is Richard Finn, and I'm a medical oncologist on faculty here in the Department of Medicine. I've been involved in an exciting new project involving a novel anti-cancer drug called PD-991. PD-991 is a small molecule which is designed to be taken orally and get into cancer cells and block cell division. As we know, cancer is characterized by uncontrolled cell growth. That is to say, cancer cells grow regardless of signals that tell it not to grow. One of the key regulatory proteins, one of the key proteins in cancer cells that control its cell division is this protein called cyclin-dependent kinase 4 and 6, or CDK4/6. The idea is that this agent, PD-991, blocks CDK4/6 function. By blocking CDK4/6 function, we can therefore block cell division. Using laboratory models here at the Jonsson Cancer Center, we initially evaluated PD-991 in cell line models of breast cancer and other malignancies. Something that we observed very early on is that in the laboratory there was a subset of breast cancers that were most dependent on CDK4/6 function and therefore inhibition or blockage of CDK4/6 by PD-991. This subtype of breast cancers, characterized by the estrogen receptor being expressed, or what we would call ER-positive breast cancers, is really the most common type of breast cancer, representing about 60% of breast cancers. What we observed is that PD-991 had a lot more activity in the ER-positive subtype of breast cancers, and we also described in the laboratory that PD-991 in combination with anti-estrogen agents like tamoxifen actually produced a synergistic interaction. What that means is that the combination of PD-991 and anti-estrogen agents gave a better effect than either agent alone. This study took women with advanced estrogen receptor positive breast cancer, and by advanced I mean it was typically beyond the breast, mostly in what we would consider incurable breast cancer, and half the women got letrozole alone, letrozole being a very standard and appropriate treatment for women with this type of breast cancer, and compared the clinical benefit of letrozole alone versus letrozole in combination with PD-991. I'm really happy to say, and very excited to say, that the results of this study are really groundbreaking and really unprecedented. First, as far as the tolerability, again, like in the phase 1 study, the combination was very well tolerated. The most common side effects, as I mentioned, were fatigue and low white blood cell count. This is something we saw in the initial 12 patients, which we also saw in a bigger cohort, but again, this was very well tolerated. A very small number of women had to stop the treatment because of side effects. And if we look at just the ability to control the disease, what we call the disease control rate, that went from 44% with letrozole to 70% with the combination. That is to say, 44% of the women who got letrozole alone had the ability to induce either partial shrinkage or prevent their tumor from growing. But for the women who got the combination, that number increased to 70%. Progression-free survival is a measurement in time from when a woman enrolls in the study to when their disease progresses. That is to say, the tumors are growing by a certain amount. What we saw in the letrozole alone arm is that the progression-free survival was about 7.5 months. With the addition of PD-991 to letrozole, that progression-free survival increased from 7.5 months to over 26 months. That is really remarkable. And this improvement of 7.5 months to over 26 months is equal to a hazard ratio of 0.37. And what that means is that there's a 63% decrease in the risk of progression of their breast cancer if PD991 is added to letrozole versus letrozole alone. The next step with this compound will be to do a phase 3 study. A phase 3 study is really the last step in trying to get approval from regulatory authorities such as the FDA. Our hope is that we will see an improvement in overall survival, that not just extending the time before their cancer progresses, but really extending the period of time that women are alive. And if we can show that in advanced breast cancer, the hope would be that we could then evaluate this in early breast cancer. As the phase 3 study gets ready to launch, we're very excited to get a very rapid accrual to the study so we can get a result from that study as soon as possible and get the right drug to the right group of patients. Thank you very much for your interest in the research here at the Jonsson Comprehensive Cancer Center at UCLA.